Welcome back to the science behind. Picture this. You buy a cup of Cryptocoryne labeled easy. You plant it, you dose your fertilizers and you monitor your light, and 2 weeks later, it turns to mush. Frustrated, you throw it out, go to the store and buy another cup of the same plant from the exact same brand. And exactly 2 weeks later, it melts again. You blame your tank, you blame the water, you blame yourself. But what if the problem isn't in your tank? What if it's in the laboratory? Today, we're going behind the scenes of companies like Tropica and Dennerle to uncover the hidden reality of how plants are made, why they are identical down to the last cell, and how that genetic uniformity might be the secret reason why your aquarium is failing. To understand why we're seeing these repetitive meltdown cycles, we have to pull back the curtain on how modern aquarium plants are even produced. We aren't talking about traditional cutting propagation here, where a grower will take a trimming and put it in a propagation bed and then let it grow. We're talking about the massive scale micropropagation, also known as tissue cultures. For major brands, traditional propagation is way too slow and frankly too messy. It carries the risk of hitchhiking algae, pests, and also snails. And in order to meet global demand, they use tissue culture plants. The process is essentially a masterclass in controlled chaos. It begins with a mother plant, which is a specimen that the company chooses for its vigor and perfect color. Technicians carefully remove a tiny piece of sterile tissue, which is called an explant, and that might perhaps just be a few millimeters long of a shoot tip or a node. This explant is then placed into a transparent, nutrient-rich agar gel inside of a sealed cup. Think of this gel as the ultimate plant comfort zone. It's packed with sugars, vitamins, and minerals. Crucially, the lab introduces also a specific cocktail of plant growth hormones into the agar gel, specifically cytokinins, which force these cells into a state of hyperactive rapid division. Instead of growing like a normal plant, the tissue forms a disorganized, rapidly expanding clump called a callus. Then, using different hormone ratios, the lab coaxes these clumps to differentiate, sprout tiny leaves and roots, and eventually grow into thousands of exact, genetically identical plantlets. It is a marvel of efficiency, and it's way cleaner than anything found in nature, and it's the only way these companies can ship millions of plants globally year-round. But here's the crucial part. From the moment the first explant was taken, every single plant in that batch is a genetic carbon copy. There are quite literally thousands of clones of the exact same mother plant, and while that sounds like a quality control dream, it creates a significant hidden biological vulnerability when they leave that sterile lab and enter your tank. Efficiency also comes at a cost. When Tropica or Dennerle mass clone a single plant, they are creating a biological single point of failure. In the wild, plants evolve through sexual reproduction. This process constantly shuffles genetic information, resulting in a population that is genetically diverse. Some individuals might be better at handling lower light, others might be more resistant to a specific pathogen, and some might tolerate fluctuating nutrient levels much better. This genetic diversity is the primary defense mechanism for any population of plants. It ensures that the species as a whole can survive environmental challenges, but in the lab, this evolution is halted. When we take a single explant and clone it 10,000 times, we aren't just getting 10,000 identical plants, we're getting 10,000 individuals with the exact same genetic weaknesses as well. This is what we call phenotypic plasticity, the ability of an organism to change its physical form or behavior in response to its environment. In the wild, a plant population's ability to survive comes from a wide spectrum of plasticity across its different members, but because your cloned aquarium plants are genetically identical, they possess a very narrow, fixed range of plasticity. Think about it, the mother plant in the lab was grown in the perfect environment. It had ideal light, optimized hormones, and zero competition. Your tank, it's a completely different world. It has different bacteria, different water chemistry, and different light intensity. The clone is trying to express its genes in an environment that is radically different from the one it was programmed for, and since every clone in your cup is genetically the same, they all attempt to cope in the exact same, potentially failing way. So when you wonder why your easy plant has melted in your specific low-tech tank, it might be because that specific clone's fixed genetic programming simply doesn't contain the tolerance profile for your tank's specific unique chemistry. And since every clone is identical, if the conditions don't match the narrow window of tolerance, the entire cup will fail, often at the exact same time. This isn't a failure of your husbandry, it's a consequence of cloning a biological system and then introducing it into a much more complex and non-sterile ecosystem. So back to our original frustration. You've tried brand A, it melted. You tried again, it melted. And now you're convinced that all aquarium plants of this species are impossible in your setup. But here's the secret that the industry doesn't explicitly tell you. Not all of the clones are equal. Just because two cups from two different companies are labeled with the same scientific name, say Cryptocoryne and Decoding, doesn't mean that they are genetically identical. In the world of tissue culture plants, they likely originated from completely different mother plants. Think of it like this. Every genotype has a tolerance window. It's the range of light, CO2 and nutrients levels that it can thrive in. Brand A's genotype might have had a tolerance window that sits further to the left. It might be heavily optimized for high CO2 and very specific soft water parameters. If your tank, your biome, sits on the right, perhaps it's lower tech with harder water, and that plant's going to struggle regardless of how much you try and fix your tank. But when you switch to brand B, you aren't necessarily buying a better plant. You are effectively swapping in a different genotype, and that clone might have a tolerance window that by pure biological chance overlaps with the specific, messy, non-sterile conditions of your tank. It survives not because it's superior, but because it's compatible. This is exactly why you see forums flooded with conflicting advice. I grew this in my little tank with zero effort versus I've tried for years and it always dies. They are both right, they are just growing different genotypes of the same species. Understanding this takes the pressure off your husbandry and puts the focus back on finding the right fit for your unique ecosystem. So the next time you see a plant melting in your tank, don't just blame the nutrient balance. Understand that your plants might just be clones, genetic siblings of thousands of others that have struggled in the exact same conditions. Sometimes it's not just about doing more, it's also about trying a different genetic candidate. Knowing that your plants are clones with narrow tolerance windows changes how we approach setup. If you've been struggling with plants that consistently melt, you might be tempted to just swear off a brand forever. But before you do that, let's talk about hardening off, the bridge between the sterile lab and your home aquarium. When you open an in-vitro cup, you are introducing a plant from a hyper-sterile, high-hormone environment into a chaotic, microbially active tank. That transition is a massive shock. The plant has been living on sugars and it hasn't had to photosynthesize efficiently because it was essentially being force-fed. When you drop it into your tank, it has to switch its entire metabolic machinery almost overnight. If you rush this, you're just begging for a melt. The best way to help any aquarium plant clone survive is to minimize that metabolic stress. First, be meticulous about rinsing the gel. It's loaded with sugars that in your tank will become an instant feast for bacteria and fungi, which will lead to an algae bloom that will smother the fragile plant. Rinse it gently but thoroughly, and use tank water if you have chlorine in your tap water. Secondly, consider preconditioning. Instead of throwing them into your main display tank, place your newly acquired clones into a small separate container, which you can consider a nursery tank with high light, shallow water, and perhaps a small dose of liquid fertilizer. This will give them a controlled environment to transition from lab mode to aquarium plant mode, without the pressure of competing for nutrients or light with other, maybe more established plants. If you've done this, if you've rinsed them, hardened them off, and they still melt, only then should you consider that maybe that specific clone's genotype isn't compatible with your tank's biome. By following these steps, you've eliminated husbandry failure from the equation, leaving the biological compatibility issue as the only remaining variable. Understanding the biology behind your plants, where they come from, how they're produced, and the trade-offs of mass cloning doesn't make you a better aquarist. It makes you a more objective one. When we stop viewing every setback as a personal failure, we start observing our ecosystems with a more critical and scientific eye. Yes, the aquarium hobby is about aesthetics. We want that perfect lush green aquascape, but it's also fundamentally an exercise in biological systems management. Whether it's the nitrogen cycle that we discussed previously or the genetic profile of the plants that we choose today, the science behind isn't just theory, it's the actual key to building a tiny, thriving and sustainable biome. We're all on this journey of constant learning and experimentation together, and if you have a specific plant or a mysterious melt story that you have been trying to solve, drop it in the comments below. I want to see if we can decode the science behind your setups as well. If you enjoyed this deep dive, make sure to like and subscribe for the next episode of The Science Behind. Until next time, keep building, keep experimenting, keep failing as well, and I'll see you in the next video. Thanks for watching.